Hello and welcome, everyone. Thank you for joining us for today's webinar, Phenotyping the Tumor Microenvironment with Advanced Tissue-Based Multiplexing Assays, presented by Dr. Katir Patel, Global Manager, Field Applications, Ultivue. My name is Chrissy Jewell. I'm with LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Today's event is sponsored by Ultivue. Ultivue is dedicated to research, innovation, and to delivering exceptional quality RUO products and services to the life science and biomedical communities. To learn more about our sponsor, please visit ultivue.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen, type in your questions into the drop-down box, and click Send. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Our speaker today is Dr. Katir Patel. Dr. Patel has extensive experience developing reagent-driven solutions for high-definition biological imaging in situ, combining high-level multiplexing at high speed with high spatial resolution, specifically in the field of immuno-oncology. Prior to joining Ultivue, Dr. Patel was a product manager for the Quantitative Pathology Solutions group at PerkinElmer. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Now, without further ado, please join me in welcoming Dr. Patel. I will now turn the presentation over to him.
Thank you, Christy, for that wonderful introduction. So I'm Dr. Patel. I'm happy to be here today, and I appreciate the opportunity here with LabRoots to tell you a little bit about what we're doing here at Ultivue. So just to get right in, we're a Cambridge-based company. We've been working with tissue multiplexing for quite a while now. We have a variety of products that are offered to the consumers that are out there today. We can engage with you in a couple different ways. One way is through our kits and our kit content, which I'll describe a little bit later in the presentation. We also have a services group that provides custom content as well if you're interested in higher levels of plexing or custom panels that fit your specific needs. So just to get right into our presentation today, the first thing we want to really sit down and discuss is the tumor microenvironment and why we need tissue multiplexing today. The tumor microenvironment is incredibly complex. There's a variety of cell types, a variety of cell-to-cell interactions within the immune system and within the immune system to the tumor. Understanding these types of interactions is key to be able to optimize and understand the best types of treatments for individual cancer patients. For example, one of the big things that we really need to keep into account, and one of the major benefits of tissue multiplexing, is being able to understand spatial interactions. So if you look on this slide that's currently being presented in the center, you'll actually see a figure that was pulled from the Bernie Fox lab at Providence Cancer Center from a publication in 2017 looking at PD-L1 within a tumor. When we look at PD-L1 alone within the tumor, you really cannot stratify patient populations in survival. PD-L1 alone was not a predictor of prognosis or outcomes for these specific patients. When you take into account spatial context with CD8 specifically, you actually get better resolution. You can actually see a stratification of patients from low to high here as far as survival goes when CD8 was taken into account in its distance to PD-L1. Now, when PD-L1 first really came out, this is one of those topics where if a patient was PD-L1 positive or negative, that would dictate the type of therapy that may be recommended for that specific patient. As we've learned more about PD-L1 with its interactions within the immune system, we've seen that now there is much more to that story than there was initially thought. Now it's really important as to where the PD-L1, for example, is being expressed. Is it on the tumor? Is it on the macrophages? And if so, what are the distances is that to specific immune cells such as CD8? These types of key interactions are things that multiplexing helps you reveal and that you can see very clearly. And being able to understand these types of interactions will allow you to better understand specific treatments that may work for one patient versus another. Another key to kind of understand with this type of dynamic is that with the development of all these new, unique types of personalized medicines or immunotherapies that are specific to very unique targets, understanding the types of immune cells that may or may not be present with each one of these cancer types is becoming more and more important. Being able to have a more unique and critical view of the types of cells that are present for each specific tumor type allows you to better understand which types of treatments may be more effective in one case versus another. And as far as tissue multiplexing goes, it's been around for quite a while. And with the current technologies that are available on the market, there's been left for quite a few things that people would be looking for to be able to really scale this type of technology and apply it to a larger group of patients. One of the big things is you want a high level of plexing. As we learn more, we need more, right? That's a very simple kind of dynamic. You want to be able to plex to the level that accounts for the types of studies that you're looking for. Some may involve, 2 to 3 plex to get the information that you need. Some may involve 10 to 12 plex to be able to get the information that you need. So you want a type of platform that allows you that type of flexibility. You also want a type of platform that takes into account the fact that samples are becoming more and more scarce and samples are becoming smaller and smaller. So ideally, you'd like to have a platform that allows you to actually use that single sample for other downstream type assays and get as much information out of that single slide as you possibly can. You also want to be having a platform that takes into account that we have the ability to do whole slide imaging now and whole slide analysis. In the infancy of tissue multiplexing, this is really limited to fields of view and smaller regions. And this is usually a limitation of just the software programs that were available. As you can see at the bottom there, as far as data analysis goes, there's a lot more options out there. There's a lot of platforms that can actually handle large data in a whole slide context. So being able to take whole slide data allows you to eliminate bias that you may have by only being limited to fields of view, because we know the tumor is actually quite heterogeneous. One of the bigger things that I think that most people have struggled with too with tissue multiplexing is in assay development. You want something that is very reliable, that's very robust, that's very easy to implement. You want it to fit into the existing workflows that you are familiar with and work with equipment that you already have readily available. Now, our UltiMapper workflow is incredibly simple. It's same-day results. It's from staining to results. So I mean, you can get on the BondRx, or you can stain this assay manually, and it's 5.5 hours end to end. You can go right into whole slide imaging, which can take about 10 minutes, depending on your scanner. And we work with a variety of different scanners. We've worked with the Zeiss AxioScan Z1, the Olympus VS120, the Leica Versa, the Vectra Polaris, where we don't actually need the multispectral aspect of it, but it was just as a whole slide imager. We've also worked with 3D Histech. So we've been able to work with most conventional whole slide scanners that are on the market. And as far as image analysis goes, this is really user dependent. There's a lot of different whole slide analysis softwares that are available, such as Definiens, Halo, etc. So again, the real thing to take away here is that we want to have same-day results. You want a simple, easy, automated or manual, if that fits your needs better, type of workflow where you can get same-day results that are reliable and reproducible. Now, taking you into our assay overview of how our technology actually works, please keep in mind that this assay is extremely simple, that we do a single antigen retrieval step, a single staining step, a single amplification step, and a single detection step for our standard kits that are commercially available. What we actually go ahead and do is we have our primary antibodies conjugated to unique DNA barcodes. Each primary antibody has its own unique DNA signature. So CD4 would have its own signature, CD8, for example, would have its own unique DNA signature. And these are very different. They do not interact with each other. They do not share any homology with anything that's in the human genome. They're very specific, and they're very unique to each antibody. So we can actually stain the slide with all of our primary antibodies in a single step, and we do a single amplification step. Now, this single amplification step of all targets is incredibly important. This amplification step is a linear amplification method. and all targets are amplified at the same rate and at the same time. And why is this important? This is important because this actually allows you to have a very quantifiable type assay, where these targets are amplified to a rate where you can really go ahead and see differential expression of a particular marker. High and a low expressor, for example, are very clear to see. And it's easy to compare markers in different channels. So you could compare your CD4 expression to your CD8 expression, for example, based on relative expression, because all targets were amplified at the same rate, same time, in a linear amplification methodology. Now, as far as staining goes, to actually detect these targets, what we actually do is we actually apply a mixture of fluorescent probes that are complementary to that single-stranded DNA barcode I had mentioned earlier. And now you're actually able to hybridize those imaging strands to that amplified DNA barcode and actually detect your specific signals. So it's a single cleared, single detection step. And the one thing to keep in mind here too as well is that this tissue is actually fully intact. This tissue is in incredibly good shape, so you can do downstream analysis with other types of technologies, other platforms, which I'll describe a little bit later. Now, with that simple type of workflow and technology I was just describing, our initial kits that we already have on the market that this directly applies to, that you can apply and run in your own lab today. These are the 4 commercially available kits: a TAC kit, APC kit, a PD-1, and a PDL-1 kit. And one thing to keep in mind is that these kits are not simply limited by the fact that they seem to be a 4-plex. They actually give you a lot more cell phenotypes than the 4 markers that are there. This is because we have an ability to do high levels of co-localization, which I'll describe a little bit later. But as you can see, we don't use any secondary antibodies whatsoever. So you're really at the limit of detection of a primary antibody. And the nice part about this assay as well is that these primary antibodies can be of the same species. They could be all mouse, monoclonals, they could be rabbit, it doesn't matter, because we do not use any type of secondary. And to kind of highlight the fact that this tissue is really well maintained and very, very much intact, you could actually do an UltiMapper staining on a single slide and actually use that same exact slide and go ahead and do an H&E on it. And now you can actually get an H&E of the exact multiplexed fluorescent slide you had just stained in a previous step. There's also, thoughts that you can go ahead and do things like, RNA-ish, DNA-fish, things like that. Other types of downstream assays. So one thing I want to dive a little bit deeper into, as described before, and just to make sure to re-highlight this, each primary antibody has its own unique DNA barcode. It's a single-stranded DNA. We do a single amplification step where we amplify that single-stranded DNA via tandem repeats. And keep in mind, that DNA amplified target is incredibly, incredibly small. This is why we don't run into any steric hindrance issues, and we can actually do high levels of co-localization. You're really at the limit of what the primary antibody can do. And as far as labeling goes, we use imaging strands that are also single-stranded that have fluorophores attached to it, but they're complementary to their specific DNA barcode. One of the unique parts of our technology is that we can actually go ahead and remove that signal while leaving that amplified DNA barcode behind. And now, why is that important? This is important because this allows us to go beyond, well beyond 4-plex and go into areas of 8, 9-plex, and so on very easily without having the need to do any sort of spectral unmixing. And to kind of go ahead and highlight that, I'm going to walk you through how you could actually go ahead and do a 9-color or a 8-plex assay on a, say, a 5-color fluorescent, 4-channel fluorescent microscope. So the steps would all still be very simple, and this is the type of offering that we can actually give within our services group where we can do these higher levels of plexing. This is not something that's commercially available for the field yet, but it's something we do routinely in-house. So you would still do a single antigen retrieval step, single DWAC step, that type of step, and you would actually apply all your primary antibodies in a single cocktail, as you can see at the top. So you'd introduce all 8 primary antibody targets at the same time. You do a single amplification step where all 8 targets would be amplified at the same rate and same time as I had mentioned before. And when it came down to imaging, what you would actually go ahead and do is you would apply a mixture of 4 imaging strands using a, say, for example, a FITC, a TRITC, a Cy5, and a Cy7 type fluorophore. These first 4 target imaging strands will actually go ahead and bind to the first 4 targets of interest in the tissue that you want to look at. So maybe your CD4, your CD8, cytokeratin, and CD20, for example. You would go ahead and now you would actually go do a round of imaging where you'd image those 4 specific markers within those 4 channels. You'd remove the slide from the microscope. You would actually do an exchange step where we actually dehybridize and remove those fluorescent signals from those first 4 markers. And now we would go ahead and reapply a new set of 4 imaging strands, except for these 4 imaging strands would be specific for the 4 markers you did not visualize in the first run. After this hybridization occurs, we'd actually go ahead and go re-image that specific slide and co-register the 2 images, and now you would have an 8-plex image on a basic 4-channel microscope without the need to do any sort of spectral and mixing. Very simple, very elegant. It's a tremendous advantage to be able to do this quickly and easily as well. So the thing to keep in mind here as well, that this does not add a significant amount of time to the assay. So if you want to go from a 4-plex to, say, an 8-plex assay, you're not going to go ahead and go for double this staining time from 5.5 to, say, 11 hours. You actually only add about 45 minutes to the end of the assay. So from a 5.5-hour assay, you'd go to just over 6 hours to get an 8-plex data. And now if you think about clinical workflows and research-type workflows for translational research projects, this is much more attainable. This is something that is much more reliable and easy to implement due to slide volume and types of hardware and personnel that may be available for these types of studies. And here's an example of an 8-plex assay that was done within our services group, an 8-plex image of a non-small cell lung cancer that I just want to quickly highlight. Just having simple workflows and, having a kit that's ready to use, that's pre-optimized, that is a big benefit. That in itself is incredibly valuable. But one of the other things that we take extremely seriously here at Ultivue is our analytical reproducibility. Is how robust are our assays, and how can we make sure that our assays are performing at the upper echelon of what's available on the market today. So, for example, what I'm showing some data here is the reproducibility of our UltiMapper PD-1 kit. What we did here is we took 5 serial sections of tonsil, stained them with our PD-1 kit, and then looked at the cell phenotype counts across each section to see what the CVs were. What you can see here for the total cell counts is that we have upwards of 1 million cells. We have just over 1 million cells here. So that is quite a few cells that we're trying to analyze, and this is tonsil to keep in mind. And what we see for CVs between CD3, CD45RO, PD-1, and cytokeratin, that we have CVs ranging anywhere from 3 to 5%. Now, to kind of put that a little bit in context, having CVs that are within 20% are considered very, very good in immunohistochemistry. Having anything that's close to 10 to 15, you're definitely doing an incredibly good job. If you're going sub 10% as far as your CVs go, that is something that you should be very proud of. That is something that's incredibly difficult to do. What we're demonstrating here is that this is a readily available kit that will give you those types of results. And one could even easily argue that having CVs from 3 to 5% across 5 serial sections of tonsil may be more representative of differences in biology from section to section to section, more so than variability in the actual assay itself. Now, having good phenotypic cell counts, that's beneficial in itself. The other piece that you really want to make sure you keep in mind when you're looking at this is that you always want to check your average signal intensity across those serial sections. And what we're showing here, and there are actual error bars there, they're just incredibly hard to see. I apologize for the resolution where you can't see those, is that you want to have good, solid average signal intensity across the board for each one of those sections. As what you can see here, that our signal intensity from section to section is incredibly reproducible. It's incredibly stable. Because you do not want to be having to set custom exposures from section to section to section in order just to get appropriate images where you can do solid analysis and do solid segmentation. If your assay is reproducible and reliable enough, this should be something that's very easily done and should be very consistent from section to section. And that's simply not where we don't simply stop there. We also add other types. We're always looking for new and innovative ways to help our customers and as well as ourselves understand the performance of our kits and understand what the dynamic range of our kits is, understand the type of performance our customers should expect from our kits. What we're showing here is a PD-L1 expression array that was created by Dr. David Rim's lab at Yale. And what this array actually has is a number of cell lines that have differential expression of PD-L1 via activation of promoters. So you're seeing a dynamic range of cell lines that are expressing PD-L1 at variant levels from high to low. 1 and 2 of the spots are negative, and then 3 and 4 are low expressors, and 5 to 8 are showing the dynamic range between those different cell lines there. And 9 and 10 are basically almost at saturation. They're very high expressors. And you can see by the array here, they're set up basically in triplicate with number 11 at the bottom being a placenta control. And this is a DAB stain showing that dynamic range. It's not always the clearest thing to see via DAB when you're, especially at the lower end of the dynamic range of which cells are positive, which ones are not, which cell lines are positive and which ones are not. Please keep in mind that all cells here actually are positive, even though it may not be readily visible by via this PowerPoint slide. So what we're showing here was a PD-L1 expression array where this was actually created by David Rim's lab at Yale, where it's a new way of actually looking at and understanding the performance of a specific assay, understand the dynamic range of what an assay can actually detect as far as PD-L1 expression goes. And these are the types of standards we're trying to bring into Ultivue now for customers to better appreciate what they should expect for performance of a specific kit. And what we're showing here is a group of different cell lines that they're all the same cell line that all have varying levels of promoter expression for PD-L1, meaning you're seeing a dynamic range of PD-L1 expression for the cells within each one of these groups. 1 and 2 are negative, and 2 through 10 show the dynamic range essentially of this PD-L1 expression. All cells within these cell lines are actually positive for the PD-L1 expression. It may not be readily clear via the PowerPoint slide, but they are all actually positive here. And what we're seeing here is a DAB stain showing that the staining pattern of PD-L1, and that the dynamic range is there. It may not be ultra clear via DAB because of the contrast. When we go ahead and look at this using our UltiMapper staining kit, specifically for our PD-L1 kit, you can actually see that dynamic range very clearly, very easily, as is a major benefit of using fluorescent-based type staining. And this dynamic range is really important because this allows you to start understanding what you can actually start to quantitate what that dynamic range is and understand what your limit of detection and what your limit of quantitation actually is for a specific assay. And going back to what I was discussing earlier, when people initially started looking at PD-L1, PD-L1 positive or negative was really what you were looking at. The more we understand PD-L1 and its interactions within the tumor microenvironment, we find out that the amount of PD-L1 and the type of cell PD-L1 is on is becoming more and more important. So having an assay that is really that is verging on the levels of quantification, by being able to set some curves, some standard curves using types of controls like this, allow you to understand the lower limits of detection and how much PD-L1 may be important for a specific treatment protocol. And that becomes more and more important as we develop more and more immunotherapies and develop these types of personalized medicines that we're seeing in recent years. In addition to being able to really have a very quantifiable assay, and being able to have an assay that's really robust where you can see this beautiful dynamic range of expression, it's also really important to note that our assays is very easily able to detect co-expression of multiple markers on a single cell. Now, that's incredibly important as well because this allows you to better identify unique cellular phenotypes. Simply using one marker to detect a single phenotype is not usually enough. Being able to look at so if you're doing a higher level of plexing, say you're looking at an 8-plex or a 9-plex type panel, chances are an 8 or 9-plex panel, you're not just looking at basic cell phenotyping. You're probably looking to identify a very unique subpopulation of cells within whatever tissue sample you're specifically looking at. And usually in order to identify these very unique cell populations, you actually need to be able to co-localize or be able to detect the co-expression of 2, maybe 3, maybe 4, maybe even 5 markers on a single cell to be confident that that is a specific cell type that you're looking at. And since we're at the basic limit of the primary antibody and we don't use these secondaries, we are very easily and readily able to do co-expression to detect co-expression of multiple markers on a single cell. So our UltiMapper kit, for example, right here, and what I had kind of highlighted with the TAC kit earlier, you can detect a variety of cell types, from memory cells to exhausted memory cells to exhausted T cells. And that's simply using, 4 markers in this specific panel. And to really go ahead and highlight that, I wanted to show this specific image. What we're looking at here is an FFPE of melanoma, and this is stained with our PD1 kit. What we're seeing in red is CD3, what we're seeing in green is CD45RO, and what we're seeing in that blue kind of cyan color is PD1. We left off the cytokeratin and SOX10 in this specific case to actually keep some clarity, because it would just kind of make it a little bit more difficult to see what we're trying to highlight here. So you have different phenotypes here, and when you have multiple colors, when you have multiple markers being expressed on the same cell, you'll get new colors. So if you have red and green, you'll get yellow. If you have red and blue, you'll get more of this pink. If you have a triple positive, you'll get more of a white color. So what we're going to highlight here is that when you're just looking around this specific field of view, you can actually see a lot of just simply CD3 positive T cells that really aren't expressing CD45RO or PD1. You can also highlight and see some CD3 positive and CD45RO positive memory T cells, which have more of this yellow color. You can also see exhausted suppressive T cells in pink, or almost a purple color, which you can see here. And then you'll have some triple positive cells that you can readily see here that are CD3, 45RO, and PD1 positive. And one thing I also encourage you to look at within this specific slide is that there are other cells that are actually expressing different ranges of these colors. So there are some cells that are actually more of the orange phenotype, where they're having variant levels of CD3 versus CD45RO expression, that they're not very cut and dry, that this isn't at saturation, where you're really seeing the dynamic range of expression from a cell-to-cell specific kind of case. There's some cells that are almost polarized, that are half yellow, half green, and kind of starting to blend. So you're really seeing that even within this tissue, you're having a range of expression of these markers from different cell to cell. So one thing I also wanted to mention too is that we're going to be at AACR this year. We do have quite a few posters that will be highlighted here, and that we encourage you to come down to our booth. It's 2048. We'll also be presenting at the Leica Biosystems Dinner Symposium on Monday, April 1st, which will be also around AACR as well. So those are all places where we would love to come down and see you and have you interact with us, and we would love to discuss any questions that you may have and introduce you to our technology a little bit further. And I'd like to leave you with one more image that I'd like to kind of quickly discuss. It's something I didn't really highlight in the previous slides just to save a little bit of confusion, but with some of our kits, you'll have noticed that we do a cocktail in our Cy7 channel, which is cytokeratin SOX10. And what we're looking at here is a cutaneous melanoma, where you can actually get 5-plex data out of one of our standard kits. Because since we put SOX10 and cytokeratin in a cocktail and they stain different compartments, SOX10 is a nuclear compartment stain versus cytokeratin, which is more of a cytoplasmic stain, you can actually very easily do a cell segmentation using pretty much any analysis software to actually differentiate these two compartments. So you can actually get 5-plex data. It also allows you to use a single type kit for melanomas versus carcinomas. And with that, I'd love to entertain any questions that you guys may have. Thank you.
Dr. Patel, thank you for that informative presentation. quick reminder to our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click the Send button. Okay, Dr. Patel will answer as many questions as time permits. Okay, our first question. Dr. Patel, what floors do you use?
So the floors that we actually use, we don't disclose a specific dyes, but generally as far as if you're going to try to configure a system to actually look at them, we use a FITC, a TRITC, a Psi5, and a Psi7 that can type filter that can visualize our dyes. And the thing to note with these is that they're all spectrally isolated, that there is no bleed through from one channel to another, so that really curbs our need to do any type of spectral mixing.
Next question. How many tests can you run per kit?
So if you're going to, with one of our kits, if you're going to run it on the Bond RX, each kit runs 10 slides, and that will be done that you can run 10 slides in up to 2 preps. If you run the assay manually, you can run up to about 14 slides, dependent on the size of your tissue. Obviously, if you have large TMAs, they'll require more reagent volume, which will cut into that total 14, but generally anywhere from 12 to 14 is pretty safe.
Now, Dr. Patel, what are the differences in terms of sensitivity and linearity if antibody A and antibody B binding ratio is 10x to 1000x? What about thresholds?
So looking at marker to marker, yes, that your binding efficiency of your primary antibodies can come into effect, but your fluorescence signal should stay standardized between the two because of the linearity of the amplification methodology. And our signal of background, regardless, is significantly higher than the background. So when you're actually doing your threshold, having very specific and localized signal is something that will be very clear for you. So typically, maybe it's not it'd be very difficult to do a very straight quantitative assay if you have an antibody that has extremely low binding efficiency. It may not be the best clone to be using in the first case. But as far as being able to detect over background, you should have no issues whatsoever.
Okay, our next question. Does Ultivue have solutions for the co-registration of multiple rounds of imaging? And there's 2 parts.
Yes.
Or is this dependent on your imaging equipment or image analysis software?
So we do. So that's why some of our technology is still brought in-house, especially for our higher levels of plexing. We do have software that we can actually do our co-registration of re-imaging the same slide and then aligning those specific images. As far as the systems that we can take, or image type files that we can take, we're pretty flexible as far as that goes. Like I said before, we can work with the AxioScan, the Versa, the Zeiss to do these types of stackings.
And Dr. Patel, how do you deal with tissue autofluorescence?
So tissue autofluorescence, that's a very good question. That's something that comes up on a very regular basis. With our assay, our signal of background is so high that you do not worry about autofluorescence. Our exposures tend to be extremely low in the FITC channel and even the Psi5s and across the board actually, that you're minimizing the amount of autofluorescence that will actually come up. So there is no need to actually go ahead and do autofluorescence removal. We've done staining of things like liver tissue, things like that actually tend to have very high autofluorescence, but the signal is so clear and so high that you do not worry about missing any positive cells.
Now we have so many questions coming in. I just want to remind our audience that any questions we do not have time to get to today, Dr. Patel will answer these via email following the presentation. Okay, our next question. How does the sensitivity compare to TSA-based signals?
So as far as the sensitivity goes, so with our assay specifically, we use a linear amplification. Tyramide-based assays do not. They use more of a geometric type amplification. And also keep in mind when you're using tyramides, they actually deposit tyramides around the epitope of interest, so which actually leads to issues of co-localization. And that when you're doing a multiplexed assay specifically, you're actually, say you're doing 3 or 4 markers, for example. You're doing amplification of each one of your tyramides at a different position at a different time. So they're not happening and they also happen at a different rate. Because as you can imagine, if you're using a rabbit and then a mouse primary antibody in each one of your rounds, you're using different secondary antibodies to do that detection and do that amplification methodology. And when you're using an amplification methodology that's not linear, you tend to run towards saturation, and it makes it very difficult to compare. So with our assay, we're extremely sensitive because all targets are amplified at the same rate, same time, and it being linear, you avoid the issue of being able to, of running into the issue of oversaturation of an epitope. So now you can truly see differential expression of a single marker on a single cell or from cell to cell. Where if you hit saturation, you're really only doing phenotyping at that point. You can't really do a quantifiable type analysis of saying one is a high expressor versus a low expressor.
What would be the minimum requirements to have a custom assay developed?
So to do custom assay development, so that's a great question as well, and custom panels. For that, it depends on the number of this is a very complicated question, but I'll give you a brief overview of what we usually need to consider. We'd also have to consider the number of plex that you're interested in doing. We've worked with well over 40 markers plus and within our services group here, so we actually have quite the library. What you really need to do is have a good primary antibody, ideally, like a clinically relevant clone or something that's been well published, something that has extremely good staining patterns. It's a good high-performance primary antibody. We'd need that in high concentration and carrier-free, and then good control tissues to really base our staining off of, and that's really where we start. So a good quality antibody in high concentration that's carrier-free.
Dr. Patel, what tumor sample types have you optimized staining protocols?
So when we actually when we're actually developing any of our kits, we actually stain a large variety of tumor types. We'll stain colon, melanoma, breast tissue, oral cancers, lung cancers, a variety of those types. We optimize our kit across a variety of tissue types under variant types of fixation processes and what have you. Keep in mind, these are all pre-optimized. So no matter what your fixation methodology was or what type of tissue you're looking at, this assay is optimized to run the way it is, right out of the box for that specific tissue type. And we also work with a variety of different cell lines and circulating white blood cells as well to run as controls. We're very large on, developing the robustness and the validity of our assay across a variety of different types of tissues and controls.
Dr. Patel, our next question, we'd just like a recap. Ultivue is a kit and a software, or does one need a specific microscope?
So Ultivue's kits, those are the commercial kits that are really available. You do not need a specific type of microscope. Really, what you need is any type of relatively run-of-the-mill whole slide scanner that can look at about 5 different colors. So that can visualize a DAPI, a FITSI, a TRITSI, a Psi5, and a Psi7. So Psi7 may be something that may be a little bit more specific to a handful of whole slide scanners that are out there, but that's really it. You don't need anything that has any sort of spectral unmixing capabilities or anything like that.
Okay, Dr. Patel, is there any additional care that should be taken while preparing slides for IHC? Are frozen sections compatible?
So we have not worked with fresh frozen samples yet. It's not to say it doesn't work. We haven't just worked with them specifically. As far as samples go, I mean, generally speaking, you want to follow the general rules of thumb when you want good fixation. You don't want to overfixate your tissues. Ideally, you want really fresh samples that were, paraffin embedded very quickly and efficiently, and they were stored under the appropriate conditions. So nothing different besides that. We don't need any other types of specific prep. Like I said, I think one of the beautiful parts of our assay is that it's very robust and it's able to handle a variety of different types of tissue and of different types of tissue quality and still give you good, solid performance.
Are there any plans to develop kits for preclinical species?
So all of our kits now are really, they're research use only, so they're really in the preclinical space. But if maybe you're referring to maybe mouse tissues or mouse panels specifically, if that's the case, there are a lot of talks that we've had about that. We've worked with humanized mice already internally with our services group, and our kit should work just fine in those types of tissues. But if that's something that you were interested in doing, our services group would be happy to take on projects that are surrounding mouse tissue specifically.
Okay, Dr. Patel, are you developing panels to other indications?
Yes, so we're constantly building new kits and new panels. So obviously right now we have the PDL1 and PD kits, which are kind of specific to a couple specific type use cases. But we also have our TACT, APC kits that are also available. You'll be seeing quite a few new ones coming out this year, so stay tuned. And ideally, we'll probably have another 6 to 7 within the year that you'll see. And they'll have varying levels of more generalized immune panels, and some will be maybe a little bit more specific, as you're alluding to.
What clones are being used for CD8, CD68, PDL1, pan-CK kits, and other commercially available kits?
Yep, so as far as that information goes, that's a lot to kind of go over. We can send you that information afterwards, so if you'd like, just drop us an email or what have you, and we can get you that kind of information when you discuss with one of our FASs or one of our salespeople. We can talk about that. But one thing I would like to mention, too, is that we take clone selection extremely seriously here at Ultivue, that when we're actually designing our specific panels, we're looking for clinically relevant clones to bring in. If clinically relevant clones aren't available, or maybe there isn't even one available out there, we do strong literature research to understand the most widely used clones that are most widely accepted through publications. We also have a very large key opinion leader network, and work with a lot of different collaborators, and we get all of their input as voice of customer to really understand which clones are ideal to use for all these specific type cases.
In addition to antibodies, do you have RNA scope probes in your kits?
No, we do not.
Okay, our next question. Have you done extensive calibration between cell culture and tissue slide results?
Yes. So defining extensive would add a little clarity to that, but we've worked with cell culture lines in some of our controls when we're doing assay development to really understand how well our assay is actually functioning and working. So if there's more to that question, I'd love to hear more about it, about what you mean as far as extensive goes, and I'd be happy to discuss that offline.
Excellent. Now we have time for a few more questions. Can your software outline a lung metastatic lesion and enumerate the intramural cell infiltration? Intratumoral, I apologize.
Yeah, so I believe the customers, if this person's asking that we did a higher level of plexing and stained the same slide, if we're trying to do a co-registration of the same slide with multiple markers, so like an 8-plex, for example, yes, we can absolutely do that co-registration without issue. If it's referring to serial sections, maybe 2 separate serial sections of that lung tumor, our software is not designed to handle that, but there are other ones that are available on the market that can do that, I believe. But I'm not sure how well those other softwares outside of Ultivue actually perform at doing that.
This one is 2 parts. How do you excite the CY7 on a Zeiss Axio scan, as I didn't believe the LED hit the peak excitation of the fluorophores? Can you swap the fluorophore, so rather than CY have a CY 3.5?
So for swapping fluorophores, that's not something we typically do. I mean, we pick the fluorophore specifically just to make sure that there's spectral isolation, so you don't have any bleed through or crosstalk. With our Axio scans, all our Axio scans here that we run are actually size 7 enabled. I believe Zeiss has two different configurations. We've run with the Spectra X, which was kind of their predecessor system that they had used to actually make their Axio scans size 7 capable. But they've actually recently developed a Calibri upgrade that they really own, that is available on their new commercial systems that are there. And I believe they can upgrade systems that are already out in the field to be able to do the size 7. So that's a simple light source upgrade.
Dr. Patel, are the stained slides archivable?
So we are looking at the stability of our stained tissue slides, and right now we've shown stability for over 8 months, stored in the dark at 4 degrees. So I'm not sure how long the archival data would be good for past that. We're understanding how long these are good for. as time progresses. I know I've also had slides that I've basically carried around with myself for 6 months in my bag, and they still scan incredibly well. And that's also to keep in mind too. It also is a little bit dependent on what type of scanner you use to visualize these. Our dyes are very stable. They're very robust, so we don't run into a lot of photo bleaching. But if you're using an older type scanner that is dumping full amounts of white light directly onto the slide while scanning, you do run the risk of starting to photo bleach, which obviously would impact the stability and the longevity of those dyes for long-time storage.
Can you dose bar-coded antibody in vivo and do analysis on section slides?
Could you repeat that question? Sorry.
Sure. It's written, can you dose bar-coded antibody in vivo and do analysis on section slides?
I'm not sure we've done that yet. It'd be something interesting to look at.
Okay, and how do you overlap the two images on slide number 10 automatically by software?
Oh yeah, so that was basically a software tool we've created internally to be able to do that type of alignment. So that's not something that's commercially available out there, but probably any serial sectioning alignment software that's out there from other vendors can probably do the same sort of thing.
Okay, and we're almost out of time. We'll have to wrap with this final question. What is the highest level of plexing you can do?
So at SITC of last year, we actually showed a 9-plex 10-color assay that we actually did staining for. Our services group routinely runs 8. If you come to see us at AACR or right after AACR, we'll be releasing some more information that might pique your interest as to what our new levels of plexing actually are. And so keep in mind, as I said before, the process that we use for our staining is incredibly gentle. It does not, degrade any of the tissues. We don't do multiple rounds of antigens retrieval. It's a single step. And that we don't really run into any steric hindrance issues, so we can do co-localization. So the number of plexing can get up there pretty quick.
Dr. Patel, thank you for your time today. Thank you for your extensive answering of our questions. I want to thank our audience also for joining us and for their interesting questions. Those questions we did not have time for today, and those submitted during the on-demand period will be addressed by Dr. Patel via the contact information you provided at the time of registration. Dr. Patel, do you have any final comments for our audience?
Just want to say thank you for taking the time to have us here today. We appreciate the audience, and we look forward to your questions. And come see us at AACR if you get a chance.
Thank you again to Dr. Katir Patel. We'd also like to thank LabRoots and our sponsor, Ultivue, for underwriting today's educational webcast. Today's webcast will be available on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us, and we hope to see you soon. Goodbye.